The topic for today's presentation is an update on Theileriosis in sheep and goats. There are three important hemoprotozoan diseases with respect to sheep and goats like Theileriosis, babesiosis, trypanosomosis, and even one rickets cell infection, anaplasmosis, has been listed under this particular economically important diseases. So coming to the introduction, so Theileriosis is a tick-borne hemoprotozoan disease, and for all these days, maybe for last few decades, this particular disease has been neglected with respect to the small ruminants. But now, because of its pathogenicity and the factors which are favorable for their establishment, presently it has been considered as an emerging disease in small ruminants. The disease is widely distributed in tropical as well as subtropical countries where ticks can survive, and hence now this particular disease is considered as an economically important disease entity with respect to sheep and goat farming. So ticks as the main vectors, they are most competent and they are versatile vectors, and with respect to the transmission of any pathogens, so next to mosquitoes they are ranked second with their vector potential. And there are two types of ticks like hard ticks and soft ticks. So especially the ticks which play an important role in the transmission of all these diseases and even in particular Theileriosis. So they are grouped under Ixodid, and they play a very important role the transmission of wide variety of pathogens, especially to vertebrate animals, including most of the viral infections and also many parasitic diseases. So hence, the disease transmitted through ticks, they are the major constraint to the small ruminant production, where they affect the health, they damage the eyes, including the skin. So let us understand a little bit about the historical background of this malignant ovine Theileriosis. The disease condition is also known as malignant small ruminant Theileriosis. And this particular disease condition, for the first time, it was described by a team of veterinarians during the year 1914, especially in Sudanese sheep in Egypt. And during those days, the causative agent of this malignant ovine Theileriosis, it was first described as Theileria ovis, and later it was named as Theileria hirsi, and presently it has been renamed taxonomically as Theileria lestocordi. So now Theileria hirsi has been taxonomically accepted as Theileria lestocordi. And especially in northwest China, Theileriosis is a very important disease with respect to sheep and goats, and for more than 100 years, this disease has been existed over there in the Tibetan region of Guangzhou province. So the first report of malignant ovine Theileriosis was during the year 1958, which was reported by a local veterinarian from a place called Sichuan province. And from that time, they established that the causative agent of ovine Theileriosis, especially in China, it was mainly due to Theileria lestocordi. But later, the phylogenetic studies as well as the biological studies revealed that this Theileria lestocordi is totally different from the other species, and they considered that in China, especially the causative agent of malignant Theileriosis is the Theileria leuvenschuni, which was earlier designated as Theileria species 1, and Theileria uhlenbergii, which was designated as Theileria species 2. Coming to this particular table, where we need to understand the different species of Theileria, their vectors, distribution, and the host. So in this particular table, 3 species have been indicated like Theileria lestocordi, Theileria leuvenschuni, and Theileria uhlenbergii. So all these 3 species are highly pathogenic, and they infect both sheep and goats, and especially Theileria lestocordi, it is mainly transmitted through Hiloma anatolicum and Physiophalas bursa ticks, and it is distributed in the Middle East, Sudan, North Africa, Asia, including India. And the second species, Theileria leuvenschuni, the tick vectors are Haemophysalis lingaensis and Haemophysalis longicornis. So it's mainly distributed in northwestern China, Tibet, and India. Then another species is Theileria uhlenbergii, which is transmitted through tick vectors like Haemophysalis gangaensis and Haemophysalis longicornis. So it is distributed in northwestern China and Tibet. And as I told you, all these three species affects both sheep and goats. And during my presentation in the coming slides, I would like to highlight about the Theileria leuvenscheni and also the work which has been taken place in the Karnataka. Again, there are three more species which have been taxonomically identified, and compared to other three species, this Theileria ovis, Theileria recondita, and Theileria separata, they have low pathogenicity or non-pathogenic in nature. So this Theileria ovis, again, this species has been transmitted through tick vectors only like Rhizocephalus, Haemophysalis, Hyloma anatolicum, and it is mainly distributed in African region, Europe, Russia, India, Sri Lanka, and Western Asia. Then Theileria recondita, transmitted to tick vectors like Haemophysalis punctata, distributed mainly in Western Europe, and Theileria separator. The vectors are Rhizocephalus evertsi, Amblyoma variegatum, and mainly distributed in Eastern and Southern Africa. There are two more species which have been reported from Spain with low pathogenicity, both in sheep and goats, and these two species have been recognized as Theileria species OT3 and Theileria species MK. But however, taxonomically these two are not accepted to the species level. So coming to the transmission of this Theileriosis. So as we all know and we are aware of that, most of the Theileria species, they are transmitted transstadially, or it is also called as stage-to-stage transmission by tick vectors. So what is this stage-to-stage transmission? So in the life cycle of the ticks, there are three developmental stages like larva, nymph, and adults. So stage to stage means if the larva has picked up the infection, further nymphal stages can transmit the infection. If the nymphal stage has picked up the infection, the adults can transmit the infection. So that's why it's called as transstadial or stage to stage transmission. And it has been shown that this, especially Theileria lestrocordi, is mainly transmitted through the tick vectors belonging to the genus Hyloma. And Theileria leuvenscheni and Theileria lestrocordi, these two species have been transmitted through Haemophysalis gangaensis ticks. And later, in 2012, Lakin and co-workers have shown that Theileria ovis is mainly transmitted through Rhizocephalus sanguineus, and also they identified one more tick species like Haemophysalis longicornis as a vector for the transmission of Theileria leuvenscheni apart from Haemophysalis gangaensis in central China. And especially in India during 1972 itself, the transmission studies have been carried out where they have shown that especially Theileria ovis, so it has been transmitted through nymphal and adult stages of Hyloma anatolicum. And later, Sysodia during 1980s itself, they have reported they have carried out experimental transmission of Theileria evertsi in sheep through Rhizocephalus species and Hyloma anatolicum. So coming to the distribution of Theileriosis and the tick vector. So in India, historically, the first observations on Theileriosis was recorded at government sheep farm, Kurikappe, that is in the Madras state. And later, many clinical studies are the occurrence, are the incidence, and even the prevalence of Theileriosis in sheep and goats have been reported from different regions like sheep breeding farm, Mahbubnagar, Hyderabad, Kameri and Shivpuri of Madhya Pradesh, Karnataka, then Tamil Nadu, Kerala, Arunachal Pradesh, Tripura, and Haryana. So when we talk about the tick vectors, we need to understand what are those characters which are actually making these tick vectors as the outstanding vectors, especially in the transmission of pathogenic agents. So it could be Theileria organisms, or it could be Babesia or Anaplasma or any other pathogenic organisms. So number one is when we look at these ticks, they have got a very wide host range. In the sense they can infest any host and they can transmit the infection. and they've got a tendency to feed on the several hosts during their life cycle. So we know that there are 2-host tick, 3-host tick, where during their developmental stages they feed on different hosts, and so that this particular character, it provides an ample opportunity to acquire and transmit the different pathogens. And secondly, they are very hard in nature, and also the longevity, they can survive for a longer periods even in unfavorable environmental conditions. And the third point is that the ticks have very high reproductive potential, in the sense they can produce 10,000 to 20,000 eggs in a batch. So the oviposition capacity is very high, so that they've got a high frequency of host vector contact. And in the beginning of the slide, as I told you, there are two types of ticks, like hard ticks and soft ticks. So when compared to the soft ticks, the hard ticks, that is, which comes under exotic group, they have got a habit of feeding for a slow period. Means the tick will attach to the host, it will be there on the animal body, then it will start feeding for about 8 to 10 days. So this time is enough. Relatively, this is a longer period, which is enough, which gives enough time for the acquisition of the pathogen as well as the transmission of the pathogen, and as well as in turn tick dispersal by the migrating or the wandering host. So many tick vectors have been identified in Theileriosis, especially in India, because we need to know this, since the distribution of a particular Theileria species is directly related to the distribution range of its tick vectors. So this particular table shows there are different tick species which have been recorded since 1950s from Raghavachari and Reddy till 2023. The next slide. So various ticks have been reported, and mainly here we need to highlight especially the three ticks belonging to the genus Hyloma, as well as Haemophysalis and Difficifalis. They play a very important role in the transmission of the Theileriosis. So even from Karnataka also, even during 2023, so Haemophysalis cujensis and Hyloma species have been reported from both sheep and goat. So coming to the life cycle, so in general, I'll not go in detail into the life cycle of Theileria. So it mainly involves the proliferation of the initial stages in the mammalian host, that is especially in the lymphocytes, and later they enter the erythrocytes, and the remaining half of the life cycle will be completed in the tick vector. So coming to the pathogenesis, so with respect to the Theileria lestocordi, as I told you in the beginning of the slide, it is highly pathogenic, especially with respect to both sheep and goats. But however, little has been known about its mechanism involved in the pathogenesis. But many reports have shown that this Theileria lestocordi, it shares most of the characters with the Theileria annulata, which is commonly found in the cattle. So where there will be pronounced pathology and high mortality will be there, which is mainly related to the development of the schizons in the lymphocytes. So there will be uncontrolled proliferation of the lymphoblast cells. And further what happens, the infected lymphocytes, that is the schizoid infected lymphocyte, so that will express the mRNA to produce high levels of pro-inflammatory mediators like histamine as well as tumor necrosis factor, and also a number of cytokines are released which are responsible for causing or inducing the fever, or which play a role in the anemia, or which cause the punched out ulcers, or even the pulmonary edema, what we see in case of Theileriosis. And further, the phylogenetic studies have clearly indicated that the Theileria lestocordi is closely related to Theileria annulata, as I told you, and it is lymphoproliferative in nature. So as I told you, there are three important species which you need to concentrate with respect to the even diagnosis and treatment. One is Theileria lestocordi and the other one is Theileria leventionii. So Theileria lestocordi, even by phylogenetic studies, it has shown that it is lymphoproliferative in nature, whereas the Theileria leuventionii and Theileria uhlenbergii, which shares most of the characters similarly, and phylogenetically they are very close to the buffali complex. Buffali complex means Theileria buffali complex means the main target is not the lymphocytes, but it is the erythrocytes. So it is assumed that the pathogenicity seems to be very closely associated with the proliferation of the intra-erythrocytic pyroplasms, which is indicated by remarkable anemia and icterus in the affected individuals. So what I want to highlight here is, when compared to the Theileria lestocordi, if the sheep and goats are infected with Theileria leuventionii, so mainly the target is erythrocytes, so it causes very severe anemia and icterus. So this is what even we have noticed during the very severe outbreak which happened during the year 2015-16 and 16-17, even in Karnataka state. So the animals were showing only high fever followed by very severe anemia and very severe weakness. So these were the three symptoms which we observed with respect to the Theileria leuventionii infection, especially in Karnataka state. So as I told you, these two species, they show similar biological characters even with respect to the tick vectors also. So morphologically or pathogenically they cannot be distinguished. They can be differentiated only by molecular approaches. And as I told you, Theileria leuventionii infection, that is the skyjoints are, and even with respect to the Theileria leuventionii infection, the skyjoints are rarely found in the host cell cytoplasm, and most of them they are found outside the host cell. Even to date, there is no explanation for this, why they are found outside the cytoplasm, but there are some hypotheses that an apoptosis related few mechanisms may be leading to release of the parasite from the host cell. So that is related to the mechanism like what we observed in case of malaria parasites. So coming to the clinical symptoms, so Theileria lestocordi is highly pathogenic. So the disease form may be acute, subacute, and chronic. And Theileria leuventionii, which has been reported to be equally pathogenic from southern and northern districts of Karnataka state. Even this has been reported from many states in India also. So where the animals will show very severe anemia. When compared to lestocordi and Theileria leuventionii, as I told you, Theileria ovis is less pathogenic and it has been reported to be very moderately or non-pathogenic. And most of the times, if the infection is there or the disease is there, it is overlooked. The symptoms, main symptoms include there will be pale mucous membrane because of very severe anemia. And with respect to the Theileria lestocordi, so enlargement of superficial lymph nodes may be there. So especially prescapular, high fever will be there, listlessness, anorexia, emaciation, increased respiratory rate, intermittent diarrhea or constipation, and loss of condition. Actually, during during the outbreak which happened in Karnataka, that is during 15, 16, and 16, 17. So in the most of the affected animals, we recorded only the eye fever, followed by anemia and weakness, but we could not see the very enlargement of the superficial lymph nodes, because as I told you, Theileria leuventionii is not so lymphoproliferative in nature. Its main target is the erythrocytes. So initially, infected animals have an apparently normal appetite, but in few days after the onset of the fever, they stop feeding and later they become progressively emaciated. So even today, the precise cause of anemia is still not known, but however, there are few reasons, like there could be some morphological changes which takes place in the surface of the RBC due to antigenic nature of the organism, or an increase in osmotic fragility, or changes in membrane glycolipid components, or it could be due to oxidative injuries, or phagocytosis due to production of autoantibody production. So these could be some of the reasons for the anemia. And when we talk about, or when we look at the morbidity or mortality with respect to Theileria lestocordi or Theileria leuventionii, so and Theileria ovis, so with especially these three species are recorded from India. But with respect to the pathogenicity, whether it is a Theileria lestocordi or Theileria leventionii. So high morbidity rate has been reported up to 100%, and even the mortality has been recorded from 46 to 100%. And even during the outbreak, whatever has happened in Karnataka also, so during that time, the mortality rate was more than 50%. And there could be icterus also in case of Theileria lestocordi as well as Leventionii infected animals. So this is a picture showing the pale mucous membrane with the slight icteric condition. So coming to the diagnosis, routine field diagnosis of ovine theileriosis is mainly based on the clinical signs or the knowledge about the disease, maybe the vector distribution and host specificity, or it's a transmission mode, or during the pathological findings observed during the necropsy examination, followed by demonstration of the parasite. But however, in the last few decades, a considerable progress has been made in the development of many diagnostic tests for tick and tick-borne hemoprotozoan disease in general. But however, the high cost and technological requirements limits the routine field application. Coming to the diagnosis as such, this particular disease can be diagnosed in the vertebrate animals also, as well as in the tick vectors also. So first I would like to discuss with respect to the vertebrate animals. So the first and foremost thing what we do go for, the first diagnostic method is microscopic examination. So even till today, the light microscopic examination has been considered as a gold standard test, and where we look for gem-sustained blood smears for the presence of the pyroplasms in the erythrocytes or the skyjon stages in the lymph node biopsy. So this is a reliable for diagnosis of clinically affected acute cases. So these are some of the pictures or the showing the different forms of the Theileria organisms which have been recorded during our study. So there are different forms like single pyriform is there, ring form, parachute form, semicolon type, nail needle type, rod form, round form, cross-shaped, or even amoeboid forms. But out of all these things, the most commonly encountered forms during a routine blood smear diagnosis is round forms followed by oval and pyriforms. But the main disadvantage with the microscopy is the level of expertise is required to differentiate the mixed Theileria infection. As I told you, with the morphological characters, it is very difficult to differentiate the species. So species can be differentiated only by molecular studies like PCR. So hence this method is useful in detection of organisms during acute cases, but it has a limitation in the carrier status where low number of erythrocytes remain infected. And the other biochemical profile are the studies like it can be carried out which reveals macrocytic and hypochromic type of anemia. So there will be decreased total erythrocytic count, then back cell volume and hemoglobin levels. And when we observe the blood smear, gem-sustained blood smears, so there may be anisocytosis and poikilocytosis, that is difference in the size and shape of the red blood cells, and polychromasia with leukocytosis. So this table shows the clear differentiation between when we are diagnosing Theileria, how it has to be differentiated from the other hemoprotozoan diseases as well as rickettsial infection like anaplasmosis. So fever, of course, it is present in all the 4 diseases. When it comes to the enlargement of the lymph nodes, so it is mainly observed in Theileriosis, and nervous signs are observed in case of trypanosomosis, and jaundice is mainly observed in case of babesiosis and anaplasmosis, but very rarely or occasionally observed in case of Theileriosis and trypanosomosis. And even abortion is occasionally observed in babesiosis and anaplasmosis, whereas it is absent in case of Theileriosis and trypanosomosis. But still, we have heard from the field veterinarians that Theileriosis, maybe in the advanced cases, it may cause the abortion. And nasal discharge, is mainly present in case of Theileriosis, because the nasal discharge, as I told you, it is mainly because of the pro-inflammatory mediators like especially histamine, which brings about a lot of secretions in the lungs, and that in turn causes the nasal discharge and even the pulmonary edema. Whereas this is absent in case of babesiosis, trypanosomosis, and anaplasmosis. And the last is hemoglobinuria. So it is mainly observed in case of babesiosis, whereas it is absent in case of trypanosomosis, anaplasmosis, and Theileriosis. But during our when outbreak took place in Karnataka, so as I told you, if it is a case of Theileria leuvenschuni, where they target the RBC cells, so the hemoglobinuria may be one of the clinical sign with respect to the Theileriosis. So apart from microscopic examination, there are other diagnostic methods like serological assays. So in this, mainly the indirect fluorescent antibody test and ELISA have been used. So coming to the indirect fluorescent antibody test, so it has been reported to be more sensitive, of course, than microscopic examination of the Gimsa-Stein blood smears. But the only disadvantage of this IFAT is the cross-reactivity between closely related Theileria species. So therefore, this IFAT may be a poor measure to assign the closely related parasite species, but however, the IFAT may be still useful for the epidemiological studies. Then coming to the enzyme-linked immunosorbent assay. So this particular ELISA has been documented to detect the antibodies against different species of Theileria infecting both sheep as well as goats. So many selective antigens have been identified with respect to Theileria species in sheep and goats, and this actually it has stimulated a lot of interest in standardization and development of ELISA as a serodiagnostic test. But however, as I told you with respect to IFAT also and even in ELISA also, the cross-reactivity is a major problem where the closely related species cannot be differentiated. But of course, this has been overcome by using a recombinant protein-based heat shock proteins, but still the sensitivity will be high, but specificity will be low. Then other diagnostic methods like molecular methods. So in contrast to conventional and serological based methods, the molecular tests, they allow direct, specific and sensitive detection of parasites. So under this, there are three DNA based diagnostics like RLB, that is reverse line blot assay. So it is a PCR based molecular genetic diagnostic assay where it can detect the pyroplasms by recognizing the species specific gene regions. The main advantage of this RLB is its reliability, sensitivity is very high, and it is very specific in identification of the different species. So hence it is a powerful tool and it is a practical assay which can and it is an assay which can be used regularly for even for detecting the low level of parasitemia, but only disadvantage is it's not cost effective. So but to carry out any epidemiological studies, the RLB will be a better assay. Then coming to the polymerase chain reaction. So this particular diagnostic tool enables detection of mixed infections of Theileria species by using species-specific primers. So different versions of PCR has been developed like RFLP PCR, multiplex PCR, nested PCR, and even during the outbreak, whatever has happened in Karnataka. So I would like to share a little bit here. So when we examined the Gimsa-Stein blood smears, so parasitemia level was very high, but we could not differentiate whether it is a Lestocodi or some other organism. So initially, from the previous studies or the literature, we understood that the only two species which are prevalent in India is that one is the Lestocodi, which is highly pathogenic, and the other one is Theileria ovis, which has got low pathogenicity. So initially when we carried out the conventional PCR with the genus specific, so it gave a very good amplification by amplifying about 1098 base pair, which is specific for Theileria genus. So what we thought, we later carried out the species specific, and during that time we just assumed that it could be Theileria, lestocordi, or oves. So just we carried out species-specific PCR for lestocordi and oves, but none of the samples which gave positive in the genus-specific gave positive for the species-specific. So then later, few of the genus-specific PCR products were sent for the sequencing, and when we subjected for the phylogenetic analysis, then there we understood and we are surprised to see that almost all the sample, that is 300 plus samples, they were showing the similar identity with the Theileria leuventionii. So our study actually, for the first time, we reported this Theileria leuventionii from India. So here the species specific as shown the specific amplicon size of 389 base pair DNA fragment specific for Theileria leuventionii. And later we carried out even the phylogenetic analysis also to understand the ancestor relationship by constructing a phylogenetic tree. So as I told you, even the Theileria leuventionii, so it showed very close relation with the Theileria buffali complex. That means to say the target is mainly the erythrocytes, not the lymphoblastocytes. So that's the reason why we observed very severe anemia in case of affected animals, especially in Karnataka state. And the other molecular study method which can be used at the field level is LAMP, that is loop mediated isothermal amplification. So it's a very easy technique and it allows the target DNA to be amplified with a high detection performance under the isothermal conditions. So the main difference between the PCR and the LAMP is, so it doesn't require any thermocycler and it is very easy to carry out. So even a drop of blood which is spotted on a filter paper serves the purpose for the LAMP. So there is no extraction of DNA, something like that. Directly the blood can be used for carrying out this LAMP. Then the other method of, or the confirmatory method of diagnosis is postmortem. So in lestocordi infected animals, the most of the changes, postmortem changes, were similar to bovine tropical theileriosis, where the carcass shows emaciation and there could be variable degree of icterus. There will be enlargement of the superficial lymph nodes. The whole carcass will be edematous, congested, and there will be hemorrhagic surfaces, and trachea and bronchi will be congested, and especially in the lungs, there will be lot of frothy exudate. So lungs, even lungs will be congested, epicardium and endocardium may show petechial hemorrhages, and liver may be enlarged with the pinpoint hemorrhages. So these are the some of the pictures, like where you can see the enlargement of the spleen and the liver and also congestion. So this is a typical picture where you can see the punched out ulcers. So these punched out ulcers, mainly it is not caused by the Theileria organism as such. As I told you, it is mainly due to the alpha tumor necrosis factor, which is released by the schizoid infected lymphoblast cells. So this is abomasal fold showing the punched out ulcers. So this is again pinpoint hemorrhages in the abomasal folds. Heart showing the hemorrhages. Then kidney cortex, which is congested, and the whole carcass is thick in nature, and heart also. So coming to the diagnosis of the tick vectors. So even as I told you, in tick vectors, so when we, especially like during our maybe field diagnosis or maybe during our investigation, so when we find some ticks on the animal body also, such ticks can be collected and they can be brought to the laboratory. In the laboratory, even the Theileria infection can be, Theileria organisms can be detected. So there are two types, like we can demonstrate the sporoblast. So to demonstrate the sporoblast, especially we need to dissect out the tick and we need to collect the salivary glands from the ticks, and just by preparation of bed count, we can easily within 15 minutes the disease infection can be diagnosed. So for this, mainly the methyl green pyronine staining is used to detect the stages like sporozoites or the sporoblast. And even further, the infected ticks can be subjected to PCR also. So DNA can be extracted, and further genus specific and species specific PCR can be carried out. And especially this particular thing, it also helps not only in detecting the Theileria infection, but also it helps in knowing the vector potentiality of a tick vector. So coming to the treatment as such, so this table shows there are few older drugs which have been used earlier like sulfamethoxine, varenyl, stectin, Deworming, so and even actiflavin, all these things. So with this, the efficacy was found to be 50 to 70%. And presently, the drugs like parvocone is being used. Buparvocone is most commonly used at the dose rate of 2.5 milligrams per kg body weight. So it has been found to be very effective for both Theileria lestocordi as well as Theileria leventionii. And one thing I would like to share here is, so during our whatever the outbreaks has happened in Karnataka state, when the infected animals were initially treated with the oxytetracycline, none of the animals responded to oxytetracycline, especially those who were the animals which are infected with the Theileria leventionii. But they gave 100% efficacy with buparvocone at the dose rate of 2.5 milligrams per kg. And even the same efficacy has been reported from Haryana also. But however, oxytetracycline can be given in the initial stages for the Theileria lestocordi, but it is totally as shown like zero efficacy with respect to the Theileria leventionii. And apart from this, even the alkaloid extracts from the stem leaves of this particular thing has been, Penganama harmala has been tried at the dose rate of 5 milligrams per kg body weight once a day for 5 days, and efficacy was found to be 65%. Presently, there is no vaccine like commercially available vaccine against any of these species. Like what we have the Rakshavakti against the Theileria annulata in cattle. So that has prompted actually for the in vitro culture of for the skyshon to develop a cell lines, skyshon to cell lines for the against the Theileria lestocordi. But however, in China, they have tried to develop vaccine against Theileria leventionii because that was a major positive agent over there. But the problem with the Theileria leventionii is it cannot establish in the cell lines under in vitro condition. So hence, only few antigens have been identified to be used as a for the subunit vaccine, but however there is no successful vaccine which is available in the market. So coming to the control as such, so there are three possible ways how you can control these things, especially Theileria infection. So one is by separating the affected individual animals and giving very specific treatment to them. And if it is always better if any species has been particular species has been identified, because as I told you, if it is a case of Theileria leventionii, so the animals will not respond to the oxytetracycline, so the buparvocone has to be given for the affected animals. And secondly, the control of tick vectors, because they are the ones which play a very important role in the transmission of disease. And the third is by increasing the specific resistance of the animals.